Hi all, my name is Vivian Tat and I'm a PhD student at the University of Texas Medical Branch. Today I'll be presenting on using next-generation sequencing in bioinformatics to explore global host responses of human lung epithelial cells to SARS-CoV-2 infection. Severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, is responsible for the ongoing COVID-19 pandemic. SARS-CoV-2 uses the human angiotensin converting enzyme 2, or ACE2 receptor for cellular entry. Therefore, we decided to use Calidrate2b4 cells, shown here, as our model. Calidrate2b4 cells are non-ciliated human bronchial epithelial cells with greater and more stable expression of ACE2. The objectives of our experiment were to use next-generation sequencing, or NGS, and bioinformatics in order to identify signaling pathways involved in COVID-19 pathogenesis and determine spatial and global host responses to SARS-CoV-2 infection. To fulfill these objectives, we infected Calidrate2b4 cells with SARS-CoV-2. After infection, we extracted total RNA at 12, 24, and 48 hours post-infection. And then we performed NGS through the Illumina NextSeq550 platform with polyA plus mRNA sequencing libraries on a single 400 million read. Following this, we used QIAGEN's Ingenuity pathway analysis, or IPA, to analyze gene expression and identify pathways of interest. Genes were included if they had a log2 fold change less than -1.5 or greater than 1.5 and a p-value less than or equal to 0.05. In Table 1, the number of differentially expressed genes are shown at the various time points, and at 48 hours post-infection, a little more than 200 genes are significantly upregulated. In Figure 1, the Venn diagram, we see genes overlapping at 24 and 48 hours post-infection. In Table 2, top canonical pathways identified by IPA are seen, and at 24 and 48 hours post-infection, interferon signaling pathways are upregulated, suggesting that the innate immune response is defending the host against SARS-CoV-2. In addition, pattern recognition receptors, which are upstream of interferons, are also upregulated. At 24 hours post-infection and at 48 hours post-infection, the top upstream regulators include interferons alpha and lambda, and this supports the findings in Table 2. This figure here shows that interferon lambda is predicted to be upregulating multiple genes at 48 hours post-infection. Therefore, through NGS, we showed that genes significantly upregulated at 24 hours post-infection remain upregulated at 48 hours post-infection, and through IPA, we identified that Calidrate2b4 cells significantly activate interferon signaling and pattern recognition receptor pathways. Interferons such as alpha and lambda are predicted to be activated, suggesting that they play key roles in host defense against SARS-CoV-2. In the future, through NGS and IPA, we aim to identify genes of interest as potential treatment targets and biomarkers. Thank you for listening to my presentation.